Okay, so for question 1, it's asking, well, it's saying there's an overexpressed BRCA1 in 293 T cells, and you need to perform a Western blot. So part A is to physically draw the blot with the proteins in the transfected and controlled cells. So I mean, be like this. We can do, so the ladder's gonna go on that side, we can do control, and then trans. And so the ladder will go down. And so the bands for the controlled cells would be kind of light. They would not be that bold. So try to draw this light. It doesn't look that much different than the ladder, but they're meant to be faint and light. And then the bands from the transfected cells would be a bit bolder. Okay. So what antibodies would you use, well, for part B, what antibodies would you need to detect BRCA1 in 293 T cells? So you need a primary and a secondary antibody. So we'll go primary, secondary, little arrow. And so well, the primary, like you could say, like mouse, anti-BRCA1. And then for the secondary, you could say rabbit, anti-mouse. And so the primary antibodies would bind first. And so the controlled would only have a couple. They wouldn't have that many. We'll draw like 3-ish. But the transfected would have a lot more. So you could draw a lot more. And secondary antibodies bind to the primary. And so again, the controlled would only have a couple binding to it. But the transfected would have a lot more. Yeah, that was part C. So yeah, the blue is blue equals secondary. Green equals primary. Cool. So part D, draw and explain the expected result from the Western blot. So the controlled and then trans. So this would only have like, it would be faint light. Trans would be nice and strong, bold. And it'd be bold because the BRCA1 is being overexpressed, and also because it has a lot more primary and secondary antibodies binding to it. And like it's in the control, it's not being the BRCA1 is not being overexpressed. And so there's not a lot of primary or secondary antibodies in it. So that would be nice and faint, but the transfected would be nice and strong. All right. Check. Question number 2, you have a cell culture line with no BRCA1 protein in it. You're gonna treat it with Taxol and then do flow cytometry. So part A is draw a cartoon for flow cytometry with like the number of plates and the basic steps. And so, actually I want to use the blue. So first you're going to want to plate your cells, obviously. And for this one, I would say you would use 2 plates. But if you were trying to get replicates for like statistics and stuff, you could do 3 replicates, so it would be a total of 6 plates. I would just use 2. So you want to plate your cells. And then I would say you want to transfect and then treat them with Taxol, so transfect and treat. And so again, having the 2 plates. So neither of the plates have BRCA1 proteins in them. And so we're going to have a control and a treated plate. So the control is not going to be treated with Taxol. So we're just gonna write C for control, but the other plate will be treated with Taxol, so we can just write T in that one. So we have a control and a like Taxol treated plate. And from there, you're gonna wanna permeabilize, which puts a bunch of holes in it, and it prevents antibodies from going in, so antibodies stay out. Okay, after it's permeabilized, I don't know. Then you're gonna wanna stain the cells. Then after that, you can visualize and read the graphs as the results. Okay. So for part B, it's draw and explain the results from the experiment. So you're gonna have two graphs. You're gonna have a control graph, and you're gonna have a transfected graph. And so, starting with the control, I think this one would just be normal, because there's no BRCA1, it's not being treated with Taxol, so it would just be a normal distribution. So it'd be the big hump at the beginning, that middle, like the line, and then a little hump at the end. That big hump being the G1 phase, the middle being S, then the little one at the end is G2 into M. So this would be a normal distribution. But the transfected would not, be a bit different. So I think it would still have like the G1 and S phase, like that part would be normal. But the hump at the end, the one that's like smaller, I think it would be the same size as the G1, if not bigger. Okay, so that's G1, S, G2 into M. And so this one wouldn't be a normal distribution. Like the cells would just kind of amass and build up at the G2 into M phase at that checkpoint because of the cell cycle arrest. And so like they would just kind of build up. And so this hump would be bigger and be about the same size, if not bigger than the G1 one. So this one, not normal.